This looks exciting. Hi guys.
Andrew.
Yeah, thank you very much for meeting me.
Yeah, nice to meet you. It's a pleasure. And Bryn.
Bryn, nice to meet you, Bryn.
Yeah, and James.
James, fantastic, lovely to meet you.
And to meet you.
And so what are we going to do?
Well, what's the best thing? You tell us. You're the
Well,
expert. Should we
let's go and have a little chat, and then find out a bit more about the Biosphere project here.
Sounds good.
And then you can explain to us about your Screams and Streams project. Okay, so yeah, basically down from the Galloway and Southern Ayrshire Biosphere, where I started a digital access project. The experiment is how can we leverage technology usefully for mental health and also for environmental education. Yeah, so it's great to be able to come and find out a little bit more about this Biosphere project in Wales from you guys. So thank you very much for having me here today.
I'm James Cass, and I help to run the Dyfi Biosphere here. We're based in the Huntleth, but the Dyfi Biosphere is a region that encompasses the Dyfi River, but then stretches further north to Tywyn and south to Aberystwyth as well. But biospheres are UNESCO designations and Welsh Government designation. And it's an area that's noted as a kind of a living laboratory where people are really engaged with living sustainably and experimenting and researching on how we can live in balance with our ecosystems, which are of course our life support systems. So we spend our time coordinating, enhancing those kinds of activities within the biosphere. And one of those projects is to find out if we are, if you like, good midwives for some of the species that come to visit us in the summer season. And so that is why Bryn's come along today to talk about one of those projects, Screams and Streams.
Thank you.
I'm Bryn Hall, so I'm the project coordinator on this project, which is Screams and Streams, which is asking the question of is there a clear connection between river health and the survival of the common swift? So every year they make the epic journey all the way up from the Congo in Africa all the way up to here just to nest. So swifts will spend sort of up to 10 months in the air without landing at all, so they'll feed, copulate everything on the wing. And then they'll fly up here, and the first time they'll touch down will be when they nest. So this is the outline of the Dyfi Biosphere. So here you can see this is our core area, and this is sort of all the buffer. And what this is here, so we've got the marked points, which is where we're testing the water quality.
Okay.
So this is where the river aspect comes into it. So we've got a good number of really engaged volunteers who are coming out to the sites with us to test the water quality here. So we've got 10 sites, so some of the major tributaries just here, and then all the way along the Dyfi. So we're trying to get a gauge of sort of if the river health is consistent across the whole valley, if there might be any issues, and how that affects insect life. So as well as testing the water quality, we're going to be doing some kit testing at some of these sites.
Cool, yeah.
To look at. I've got a little sheet here, which is identification for all the macroinvertebrates that live inside the river.
You mentioned the core area, and the core area that it looks like you're trying to protect is an estuarine area, not a land area.
Yeah, I think James is probably slightly better to pick this one up. But
Yes, I think that's to an extent it's true, but every biosphere, when it's set up, when the designation is set up and when the management plan is agreed, you have to have a series of strictly conserved zones, so nature reserves and SSIs and things like that. And at the core zone for the Dyfi Biosphere is this area here. I think the hatch line there is just denoting the area of sea and tidal area.
Okay.
But our core areas are the nature reserves at Ynys Las, Ynys Here, and the other nature reserves around that area.
Okay, so we've just been to Ynys Las this morning before we came here, and we're looking at the samphire grass in the sand and the salt marsh there. And we're looking at the little cockles in the little pools. We didn't really go onto it. We just kind of looked from the edge, and we're looking at there's some nice there's a rose I'd never seen before, kind of creeping along the dunes. First of all, I thought, oh, that looks like a dog rose or something, but it's not. It's a very low rose, like a little miniature rose, very nice looking thing, and lovely orchids out there as well at the moment.
Of course.
So yeah, we were talking a little bit about sand dune habitats and you know, the importance of them. But yeah, if you have any other information about that, that would be interesting.
Yes.
But sorry, I don't want to stop you on your explaining the Swift project. So yeah, so let's go back into that.
Yeah, so we've got the 10 key sites where we're testing the water quality, and what we're also hoping to achieve is to get people throughout the valley also surveying swifts, so letting us know when they see them, where they see them, and you know, vitally where they're nesting. So we know if, say, there was some sort of discrepancy with the river quality in one area, whether there is some correlation there maybe between the swifts that are nesting or feeding and where they are or aren't able to do that. So what we're really hoping to do this year is get a baseline set of data that we know this is what the valley is currently at, and then year on year we can build on that and we can see if there is a change, if the river changes or swift numbers change, and see if there is any connection there. So yeah, we've got loads of volunteers sort of throughout the area, but we're really trying to spread the word to get as many people involved as possible.
When I arrived in this area, the noise of the swifts is really, really quite distinctive.
It is.
And so do you find that people engage because they really have a kind of connection with the swifts, because they must have heard them all their lives if they live here and that sort of thing, you know? It must be almost like a kind of like a seasonal indicator almost.
I think so. We had so someone on Sunday who came to one of our events saying, you know, for a lot of people summer starts when the swallows arrive.
Yeah,
but he
right.
was saying for him it's when the swifts arrive.
Yeah, cool.
Because they're so distinct, you know, they fly through the sky at, you know, well, I think the latest recording was 75 miles an hour.
Wow.
So, you know, incredible speeds. It's just a shadow that goes past, and when they gather in numbers and they start their screaming parties, or they're really screeching and flying around, you know, it's very distinct.
Yeah, it is, yeah.
You know, it's really sort of quite emotive, isn't it? It's
Yeah,
Screeching
that's a
sound
great
of
word.
summer.
Yeah. So sorry. So where are you planning on doing your kick testing with the with this equipment here?
Yes.
So this all looks very like you might be using it for kick testing.
Yes, so we'll be doing that at the 10 sites that we've got throughout the valley, so where we're testing the water quality. I don't know if your audience have ever partaken in any kick testing.
We've never done kick testing on stream. We haven't even done pond dipping on stream. Got a bigger version of this net, but it's exactly the same style.
Yeah. Yeah, it's a brilliant activity. So basically you kick up the base of the riverbed and you catch whatever, so you hold this downstream and you catch whatever gets into there and then you pop it in a tray, and you can pick out all number of different species that you just wouldn't necessarily expect to be there.
Yeah, so how do you work out the water quality based on the biological indicators that you're talking about then?
Yeah, so there's a lot of macroinvertebrates that live in the riverbed are particularly sensitive in comparison to other species such as fish, which are more transient, because if there is a pollution vent or some sort of issue with that water, they have far less ability to move away. So they tend to be quite vulnerable to sort of pollution events and any sort of little strange things that shouldn't be happening in the riverbed. There's also particular species of macroinvertebrates which are especially sensitive to pollution, more so than others. So by picking out these species, putting them in the tray, and then recording how many there are and what species there are, you get a good gauge of what that river health looks like. And it's especially helpful for comparing across the river basin, because you have that direct comparison.
Yeah.
And you'll also see there's a change from the tributaries to the main estuary as to what species you get.
Yeah.
Based on habitat.
So just to feed back to you what I heard you saying, so it's the types of species and then the numbers of those species. So you might have lots of species, but you might have an abundance of a particular species, and that might indicate certain, I want to say, not to say quality of the environment, if you like, but it may be, from what you were saying, it may be like a kind of more localized quality. It might not be necessarily the whole stretch of river or whatever.
It can be, and especially because of the nature of the riverbed that we're testing, you've got lots of tributaries coming in, So if there's a potential source of pollution or whatever else, it could be coming from a particular estuary and affecting that particular stretch more so than it would up or downstream a lot further.
So what sort of things pollute rivers? What sort of events, you mentioned these events, how do they occur? What are they?
There's a range of sort of different potential events. I mean, the big one that a lot of people have seen in the news recently or in the last few years would be sewage discharge.
Okay.
Because the nature of our sewage systems, when there's lots of heavy rain, often they don't have the capacity to cope with processing that sewage, and it just gets discharged before properly being treated.
Okay.
That can be a big impact.
And does that wash away into the sea, sort of thing, with the flow of the river, or does it sort of settle in some way and then affect on a more long term?
This is the really challenging thing with river systems in general, is because they're constantly moving. What could be a potential source of pollution at one point, you might test it at that point and find it, but if you test it a few hours later, because it's constantly moving, you might not necessarily detect it, which is why this kit testing is really important. Because it will affect those species for a lot longer than it affects the actual water itself.
Yes,
So what we're hoping to do then, so in the next few weeks, we'll be getting volunteers out to help us with kit sampling. So we'll be going to each of the sites over a number of days, and then we'll stand into the river, we'll go in with waders, and we'll kick up the samples, and then we empty them into the tray. So you do a timed kick so that each site has a comparable amount of time spent kicking up the dirt. And then what we do is we use one of these guides to help us identify all of the various macroinvertebrates that will hopefully be in the river. And then with that, each one of these, with the scale that we're using, has a sensitivity score of how much sort of pollution, or sort of how sensitive to pollution they are. And then from that we get a comparable metric that we can compare from site to site, so we can see if any of the rivers are sort of especially species diverse or especially not species diverse. And that will allow us to really get a good picture of how the river health at each site is affecting our macroinvertebrate life.
Okay, so how do you link that in with swifts?
So swifts, I've got another sheet here. Swifts primarily feed on flying insects, and there's various habitats that support flying insects, but a lot of insects, especially mayflies are a really good example, dragonflies, they start off life underwater as fully aquatic insects, and then after a certain amount of time they will come out of the water, they pupate, and they become a flying, fully terrestrial insect. And then they often very quickly get eaten by flying birds. And swifts are one of those birds. So river systems are vital for all life on land, but it's believed that they are especially vital for birds like swifts. So we really want to be able to identify how clear that link is, and whether the health of the rivers is making an impact on the unfortunate decline that we're seeing in swift numbers.
So what are the instructions then?
Yes, so this is a little sheet we've put together, which is how to survey swifts. So what we're using, if you're based up in North Wales, is there's a data input system called CovNod. And what that is it's a North Wales sort of ecology data input system. So any species that you spot, you can log them, and then it's used openly accessible data that can be used by researchers or the public to see what there is. And then there's also, this is a UK-wide app run by the RSPB, which is Swift Mapper. So you can download an app and you can record screaming parties or nest sites. So we're really encouraging people to use both of those apps, and that's where we'll collate our data from. So our method is fairly simple for this. It's spending sort of a minimum of 20 minutes outside around your local area, typically towards sunset or sunrise, where the swifts tend to be most active. If you see any flocks or any, especially screaming parties flying near houses, or if you see nesting activity, which happens very quickly, it's very hard to spot, then making sure that you record that on one of those two databases. We've got, if you can, again very difficult, but taking a picture of them as well, because it just helps to identify it definitely is a swift.
Yeah.
Because they're quite similar, at least aerially when you're looking at them.
Sure.
They can look quite similar, quite easily confused with swallows and house martins, which have this sort of like similar sort of shape.
I'm just going to zoom into that picture there.
So we've got the swifts just here. So swifts have this sort of sickle-shaped wing, really long wing. They're completely black underneath, quite sooty colour. And they have a very, often quite hard to spot, but sort of a light patch on their throat. Swallows and house martins, on the other hand, have these sort of like lighter white undersides to their bodies. So that's one of the easiest ways to tell the difference between them, alongside this wingspan. So it's just making sure that sort of everybody's able to sort of like easily identify them, because once you've spotted them once or twice, you know they're very distinct.
Sure.
And yeah, if there's any changes in what people are seeing, so you know there's 10 swifts flying over your house every night, and then one night there's 15, making sure you sort of log that change, or even if there's a sort of like a big decline in that.
Yeah.
Just so we know how those numbers are changing and what could be influencing that. And then yeah, there's just another map of the area that we're testing. So it's quite a nice, straightforward method.
It is. Yeah. And so it's something that anybody can do.
Yeah, absolutely.
Yeah.
Especially with the Swift Mapper app. So all across the UK, that's really widely used. And yeah, it's just really easy. If you spot them, you just pop it in the app, and yeah, nice and straightforward.
Yeah, that's great. That's really good. So, are you doing a survey today?
Yeah, so we're actually going to be doing a river testing service today, just upriver in Chemice. So that'll just be testing the water quality. But you're welcome to come along.
Where's Chemice?
So Chemice is pretty easy to show you on the map. So we're just down here. This is Faintsignal where we are.
Yeah.
Chemice is about 5 or 6 miles upstream.
There's been quite a lot of testing going on. We're working with the new Dyfi Fisheries Association, which is the strangely actually it's a fishing association, but they are the landowners. They own the bed of the river, which is normally fishing associations own the rights to the banks of the rivers, not the actual riverbed. But in this case, they own the riverbed, and so they're very interested in what's going on with their river and how that affects the sea trout and the salmon as well. So they are working in partnership, in full cooperation with the Screens and Streams project, but they're also working with the Natural History Museum in London doing an environmental DNA study. So they are pulling deep sediments out of the river in order to, shallow and deep sediments out of the river in order to do an analysis of the DNA that's captured within those various different levels.
Doing like a core sample sort of thing, I suppose, or something like that.
Well, they describe it as pulling water through the sediments.
Interesting.
And so rather than it being the kind of core sample that you might do in a bog or in soils on land. But I haven't actually seen it being done myself yet, but hopefully we're going to be there when they do the next
It sounds very interesting, yeah.
But the interesting thing about that is that it can tell you not only which species are alive and present within the river at the moment, can actually give you quite a lot of historical analysis, we're told, as well, going back up to 100 years to give you an idea of what was there 100 years ago in comparison to what is there now. And so with that that's an example of how technology and genetic analysis can give you a really interesting, as Bryn was saying, we're doing a bit of we're setting a bit of a kind of a baseline survey at the moment. But it's interesting now that DNA technology can bolster that by giving you, very likely baselines from periods in the past as well. The reason that there is a biosphere here is that because since the start of the environment movement and the establishment of the Centre for Alternative Technology here in this valley, there has been a huge amount of people who cared about the environmental cause have flocked to this area. People with really visionary ideas, and they've carried out. They've found this valley to be a place where they can run their projects, and so there's a huge amount of historical projects that have happened here in the Dyfi Valley, which mean that it's seen as being a leading area an area that has a real concentration of sustainability and environmental projects going on here. It might just be worth saying as well that this is made possible by a big project in the UK called LPIP, which is the Local Policy Innovation Partnership, which is I suppose it's that way of it's a way of taking all the great work that's done by academics in the universities and all the enthusiasm that there is on the ground from people who really want to make the environment work better, to move back into, at least into balance with our ecosystems, life support systems. And what this particular bit of funding does, it works all over the UK, but the bit that we're involved with is the LPIP that's from rural Wales. But it allows us as the Biosphere to bring together the enthusiastic volunteers that we knew were out there. Some of them were already putting lots of swift boxes into place throughout the Biosphere, and others were already testing rivers throughout the valley. It allowed us to bring those together, bring those two together, and put extra resources into what they were doing, provide them with a good high quality kit, make sure that everything is being recorded really well in a way which means that it can be available to everybody, to the public, but it's also done in a format that's useful for academics and students studying the effect of human activities on our ecosystems. And but then eventually, all of that, none of that is particularly useful unless you can get our elected governments to
act on
change
it.
policy as well.
So
a large part of this project at the beginning was looking, kind of backcasting and saying, okay, who do we want to influence? Well, we want to influence the Welsh government to make sure its agencies are looking after our rivers and keeping them as healthy, natural, wild places. And so how do we do that? Well, we do that by finding out from we have academic partners that are part of this that we meet once a month as well, and they provide us with specialist knowledge and guidance, and they're helping us to write the final report as well, so that it will be written in a way, or so the data is collected in a way, and it will be written in a way that means that it can add to that useful sum of human knowledge about swifts and river health. But also, what we want to do is we want to storify it as well. We don't just want it to be useful to academics. We want to be able to influence younger generations and that general public out here. So we're hoping to create the report, but then create a more, if you like, pictorial, storified version of it as well that can go out in both languages to the schools in this area. As well, to tell them the story of this particular environmental project that happened in summer 2025 when the swifts were in the valley.
There we go. Yeah, you just got to be careful then if the river's really full.
Yeah.
And quite fast flowing. So we've got a various range of different bits we're testing for. So we've got a little paper sheet, but we submit it all through an app afterwards. This is our testing equipment. So we've got those. Well.
So is this nitrates and nitrites, pH sort of things?
Yeah, so we've got pH, and these are the nitrates and nitrites.
Yeah.
In here we've got dissolved solids, and electrical conductivity, as well as oxygen.
Yeah, okay.
And then this one is our nifty little phosphate tester.
Okay, cool.
So we'll probably start with this one, because this one takes a few minutes to sort of run through
generate,
the process.
yeah.
So what this one is testing for is the amount of phosphates that are in the water system, which are just a nutrient basically. So if you've got too many phosphates, then you can end up with algal blooms and various other issues. Did you want to have a go at filling one? I'll show you what to do.
Yeah, sure.
So what you can do, well, we can rinse our hands. I've got hand sanitizer for afterwards. To dip it in, give them a little rinse around first, and then you fill it up. There's a little white line just there. You fill it up to the white line, and you'll see there's a little meniscus there. And you want the sort of central part of the meniscus to be level with that.
What's a meniscus?
So a meniscus, I don't know if you can see it in the
Let's
camera.
try and zoom this in. I can zoom in right tight, so.
Is the little sort of dimple you've got there where it's sort of inverted downwards.
Yeah.
And that's just to do with the shape of the bottle. Essentially, it just
That's difficult. Now that's better. Oh, that's weird. I was trying to get zoom in. Bring it closer to the camera. That's it. So the meniscus is this kind of curved surface.
Curved part there.
Yeah. Okay. Yeah, we're testing the water Korg. So Korg 909's got a pond in his big pond in his back garden, and he's put some carp in that he had reared, and he did a sort of transfer from the tank to the pond, but he's had a few losses.
Oh, okay.
So he's not sure what's going on with that.
interesting.
But yeah, we're testing the water Korg.
So what we can do now, so basically how this works is it shines a light through the liquid. So,
yeah.
So what we'll do is we'll put a clear one in, we'll clean the bottle, and put it in, it'll shine the light through, and it'll just get a reading of how much that light is sort of diffracted.
Yeah.
And then when you add the reagent in, which is this sort of powder stuff, it reacts to the phosphate in the water. So
Okay.
what that'll do then is it will increase the refractive index, which is the amount the light is scattered. And then that's how it determines the difference in phosphate.
Okay, so the water's being tested because they are running a citizen science project where they want people, members of the public, people to basically test water quality to see. They're trying to work out if there's a correlation between water quality in the area and swift populations. So the science project is called Screams and Streams.
So what we're doing is we're just opening up this sachet.
Yeah,
There's a bit of a knack to it because it's windy. We're doing it inside the tub.
that's clever. Yeah.
And what you want to do is you want to get a proper little sort of funnel down there. And then what you can do with that is you can just pour it into here, hopefully without making too much of a mess. There you go. Just a little bit left in there. If any does get in your hands, just brush it off.
Yeah. You don't lick your fingers afterwards.
No. Oh yeah. We certainly did. Here we go. Pop the cap back on then. So we're just testing for the phosphates.
You shake it or?
So what we'll do now is we do a shake of this until it's all dissolved for about 2 minutes.
Mm-hmm.
So I'll put a 2-minute timer on. There's a lot of timers with this.
Yeah. I've done similar things, but with bacteria counting.
Ah, okay. Sort of familiar territory then.
Sort of, yeah.
Right, so what we do with this first then is we want to calibrate it. Again, we'll do this in the tub so that it doesn't get blown around all over the place. So what we do is you just turn it on, do one tap, and then it'll come up with C1, so that's calibration. You've got to make sure there's no sort of water or finger marks or dust on this. Here we go, clean. Put on that. And then we press and hold. We just tap for the first one, and it will calibrate.
Okay.
Just 3 little dotted lines.
Yeah, we're seeing that. Yeah.
Sometimes it's very quick, and sometimes it's not. No, Stuart, sorry.
No problem. See you again.
Lovely, there we go. So it's ready for calibration 2, so I'll just pop that open slightly. How's that looking? Oh, it's getting there.
Yeah. What are you looking for when you say it's getting there?
So there's
Changing.
yeah, we just put this reagent in, so it's just seeing it should dissolve into it,
Once it's dissolved, yeah.
and then it's ready for testing. Yeah, you'll see if there's ever really high phosphates. Already this is looking somewhat promising. As if there's really high phosphates, it actually changed as the colour of the liquid, and it will go sort of a bluey purple colour.
Yeah.
So you know if the phosphate's going to be particularly high before you test it. So that visual indicator.
Mm-hmm.
What are we looking for here? High or low phosphates?
You need a certain amount of phosphates in your system because they're nutrients, so they're
Yeah,
really
yeah,
important.
yeah,
But yeah, we want low ideally.
All time
There we go. How's that looking? Is that pretty much dissolved? pretty much there. Give it a little flick, because I think there's just some air bubbles in there. Get some of those out. Yeah. Take this one out. Look at that. And then we can just give that one a good wipe. Yeah, that's good. That's dissolved. Pop that in there. And then you press and hold this one. And it'll come up with a 3-minute timer. There we go. And then you just leave that for 3 minutes and it'll sort itself out. So we can leave that one for now and then start on one of the other things. So next one that's quite nice to start with is the pH. So these ones we're just using these little dipsticks. So did you want to go with that one?
Yeah, sure.
So these ones, I'll read you through the instructions. 2, I think.
Is it litmus paper sort of stuff?
Yeah, that kind of thing.
Yeah.
So what you do with these ones, so you'll dip it in for 10 seconds, or we'll just grab another one.
Yeah. Sorry.
That's alright. Just put that in the tub. There we go. So you dip it in for 10 seconds with a gentle movement.
This way?
Coloured way. Yeah, coloured way down. And then I'll start the 10-second timer on that just so we're nice and act through it. There you go. So you're going to go now. So yeah, just gentle backwards and forwards movements. And then once that's done, so 3, 2, 1, there you go, you take it out, 1 brisk shake, and then we will count for 20 seconds. And then once that's up, what you want to do is you want to match it to this. So the bottom line, so that sort of more greeny one, should match up to one of these colours. 3, 2, 1, there we go. So if we hold it up like that, so that's probably about
It's kind of in between.
Somewhere between 6 and 6.5. Yeah. We can make a call with that one. So is it? It's probably a little bit more on the 6 side, I think, isn't it?
Yeah.
Yeah, it's more on the green side of things.
Do you think more on the green?
I know the
like-
top feels more there, but the bottom feels more in 6.
Yeah, exactly.
Yeah.
It's kind of diagonal,
Yeah.
isn't it?
Yeah.
Yeah.
We'll go with 6 because I think it's a
Yeah.
little bit closer to that there.
Can I show the audience those two things together, just because it was quite far away?
Yeah, there you go. It will
Thank
change
you.
colour now a little bit.
Yeah.
Just as time goes on.
So what we were saying there is that it looks a bit like
So
the orange looks a
just
bit
got
like
this.
that
Yeah.
orange, and
6, 0.
it's not the. If you look at 6, it's kind
Did you want to go with the?
more orange, and it's not. The next one, 6.5, is more green.
Yeah.
So it's changing and changing as we go here. So it's kind of like that. So that's,
Oh, okay, yeah.
and that's the pH.
Oh, okay, brilliant. Let's go with that one then. So this is the.
Do you want to keep a hold of this?
Yeah, if you'd like to just pop it in there. Thank you.
Sure. Thank you.
So this one's ever so slightly different. So this one you dip in for 1 second, so just in, 1, out. And then don't shake it at all, so try and shield it from the wind.
Okay.
And then we'll wait 30 seconds and check it then. There we go. So you go when you're ready. That's 1. And then we wait 30 seconds. And it's the same again. We've got to compare this one quick. You've got about 10 seconds, I think, to line it up. And what we're aiming for. So this is nitrates and nitrites. Again, nitrate is a nutrient, so it is needed in a water system. But again, too much can be a bad thing. Nitrites are basically toxic to everything.
Yeah.
So you don't want any of those. 2, 1. There we go. And that way again. Normally these, because they're that sort of litmus paper type thing, it normally comes up 0.
Yeah.
Realistically, it's going to be a little bit more, but what we're just looking for really is just
No
that it's
pink.
not.
Yeah,
Yeah,
sure.
it's not really high. So we put them both down as 0.
Yeah, thank you. Thank you. So
The phosphate is
there you
done
go.
there.
You can see there.
So the phosphate has also finished.
Okay.
Back to there.
Sure.
That one is showing a 0.00.
Okay.
What we do when it's on that score is that we tend to do it then twice, just to be sure, because it's very rare that you won't have any phosphates. But these checkers have a slight error margin of 0.04 parts per million. So the likelihood is that's somewhere between that and 0. But we'll do another test just to be sure.
Slytherin Granny is saying, if pH is 6.5, isn't that okay-ish? Thinking about aquariums here.
Yeah, so for most water bodies, you know, like the water is naturally, when you don't take into account bedrock, would be 7, that's neutral. 6 and 6.5, that's getting towards acidic, but it's still not really acidic as such. So yeah, that's healthy enough for any river creatures that might be living.
Yeah.
But it's largely affected by, it can be affected by forestry and things like that. It can also be affected by bedrock.
So you're talking, when you say forestry, it's like the acidification from pine.
Yes.
Basically, yeah.
Yeah. Yeah, soil conditions.
Yeah.
Play a really big role in it. You also find peat bogs as well. They tend to be quite acidic. So we'll just do this one again. A bit of a nightmare lining these up. Oh, there we go. Would you like to hold it again? Yeah. Pour in.
So how much do these kits cost?
They vary. So the phosphate checkers, for what they are, they're not too expensive. They're about sort of £60 to £70 normally. These sort of little litmus tests, they're fairly cheap, so you get sort of 50 to 100, normally around £10–15.
Okay.
With the other kit that we're going to come on to, the oxygen testing, those are a lot dearer. They're normally a few £100 for sort of quite basic ones, and then
Yeah. So when you say the kit, that includes the reader and everything like that, or is it just for refills for?
Yeah, so the actual reader itself, you'll normally get sort of 3 to 4 tests with it, so 4 of these pouches.
Yeah.
And then you'll have to buy additional ones.
Yeah.
100 of those, normally buy them in quite big packs like that. You're probably looking around 30 to 40 quid.
Yeah,
depending,
okay,
30 to
yeah.
40 pounds.
It's particularly if you've got to do double testing and things like that, it
adds
becomes
up.
quite expensive, I suppose.
Yeah, you always hope you don't have to do the double testing. So it starts to put a bit of a price tag on it. But,
It is really cool, isn't it, Red Robin? I have to say, I'd like it as well. They're asking where your backpack's from.
a backpack.
They like your backpack.
It's a Fjallraven.
It is
backpack.
a what?
A Fjallraven.
Fjallraven.
They're,
Is that like Norwegian or something? or
Swedish.
Swedish.
It's that's the logo.
Oh yeah, with a fox.
For anyone interested.
Yeah, it's cool with is it a laptop bag or something with a big mouth at the top?
It's not really designed for a laptop, but I've got a laptop case in there.
It's just because it's got the
padding,
lovely
but-
big square, like open top like that. That's
Yeah,
it's a little bit like a dry bag, isn't
yeah,
it?
it's meant to be a roll down, so it's not fully
interesting.
waterproof,
Yeah.
but it tends to do the job.
There you go, Red Robin.
Here we go. So
2 minutes, isn't it?
You can look it up
so
now.
if you start shaking again, do that 2 minutes. There we go. What's that next one then?
So they were just discussing the tests there, Red Robin, and so they come. The equipment is more expensive depending on which test it is. So the pH and nitrite tests are the cheapest ones. The phosphorus test. Was it phosphorus?
You went for that one, Tyler.
The first one, the one in the green, is it phosphorus?
Phosphate. Yeah.
Phosphate, yeah. So phosphate tests a bit more expensive, about 50 quid for the kit, and then the refills for the putting into the water, and then now there's an oxygen tester, I believe.
So this one is electrical conductivity
electrical conductivity.
total dissolved solids.
Oh, cool.
So it'll start off with the total dissolved solids. You can see parts per million on there, and it's worked out on a scale based on the electrical conductivity, which is what that 0.7 means. And then we've got temperature as well on the bottom there.
Okay, and does geology affect conductivity as well?
It can do, yeah. Certain things like heavy metals, soil, various things affect electrical conductivity.
Yeah.
It's the sort of scientific side of it, is it's the total dissolved salts essentially in the water. Some things, so if you had an oil spill, that would reduce your electrical conductivity. Certain metals or heavy metals that are washed in from like old mines, those could increase it, or things like lead again could decrease it. So you've got various factors that can play a role in your electrical conductivity.
Oh, interesting. Very interesting.
Here we go. So if you just pop it in and just swirl it around, this one takes quite a while to...
Here we go, you didn't tell me that before I tried it.
Yeah, we have another volunteer sneakily snuck in behind.
So you can use the same water again, like, you know, because you're kind of putting the paper into this bucket, it's alright?
Yeah, because there's enough there, it won't make any difference to it. So we're just resetting this one again.
Get some swirly action in here.
You never confirmed how long I was doing this for.
So if you can see the numbers,
I think
just
you meant
there.
to do it clockwise, aren't you?
It's only in the southern hemisphere.
Yeah, right.
Do you want to start giving that a wipe and give it a little swirl again? Loads of little bubbles in there. There you go, you're going to give it a wipe and when it's ready for this. In that. There we go. And then so it's on C2, and when it's on that you just press and hold until it comes up with 3 minutes. Let's see what that gives us. Yeah, so this one does take a few minutes to give us a reading. And what it typically does is when you write the reading down, it'll then change. And you have to write it down again. Okay, so we can probably take the temperature reading from that now. Oh no, that's still changing. We'll give it a little bit longer.
It's changed quite a lot, didn't it?
Yeah, if you go ever so slightly slower, there you go.
Like porridge.
Yes.
Is it the same for this one as well?
It's the same process, yeah. Yeah, so this one has, so this is the dissolved oxygen. And at the bottom here it has really tiny like micro holes. That slow moving motion allows the oxygen to enter, and that's how it gets its reading.
Seems to have settled on 15.9.
Seems fairly stable, doesn't it? So you've got 15.9. And then it used to be on 33.3 as well, doesn't it? So if you keep it in the water for a second, so 33.3, and then on that bottom button, so keeping it in there, if you press in, you do it for you, and then we just change it around to the electrical conductivity, which is just that. So that seems stable as well. So 51.5. There we go. Oh.
As you wrote.
So you put that one out. If you just give it a little shake, and then you can put the cap back on and just switch it off. Him again.
Just try that off? Or?
Yeah, just pop it on. I'll clean it up when we get home.
On and then.
Yeah. So what you've got to do with this one, what it's currently reading, it's got dissolved percentage of oxygen. Yeah. If you press and hold that middle button until it changes, it should change to milligrams per litre. There you go. And then so same process again, just that gentle sort of moving around. If you just submerge the, you see the little black dot there?
Oh yeah.
Just make sure that's submerged as well. That's reading the temperature there.
So, Bryn, you're being asked, do you do this trial in Wales, only in Wales, or all over?
So yeah, this is part of the project, which is Screams and Streams project. So we're only currently doing this in the Dyfi Biosphere as part of this project. But a lot of this is very sort of standard testing procedures that they do throughout the UK, various organisations and charities that do similar things. So yeah, as part of this project, we're just in Wales, so we're on the Dyfi River. But yeah, we're not outside of this area.
Amazing, thank you.
You're going to be using this data to make a prediction on the kick testing when you do it? Or?
Yeah, so when we do the kick testing, it's going to be really to see So this is giving us a baseline idea of what these rivers are like, how healthy they
Yeah.
look, and then when we do the kick testing, it'll give us a sort of a clearer picture of how or whether that's affecting the biodiversity.
Yeah.
Yeah.
But this should give us an idea. So there's 1 or 2 sites which we're testing where we are getting much higher nitrates and much higher phosphates, and the turbidity is higher. And you could predict that there's probably going to be a little bit less diversity there in terms of macroinvertebrates.
Yeah.
But until we do the kick testing, it's not necessarily possible to guarantee that.
Yeah.
But it's a good indicator for it.
Somebody in chat was saying that they thought that mayflies play a big factor with swifts. They think is what they said.
Yeah, so it's part of sort of why we're doing this project. So we're looking to see if there is that clear connection between swifts' survival and the health of rivers. And mayflies do start off their life as larvae or nymphs in the river system. And typically mayflies can be quite vulnerable to different pollution, different things that are coming into the river. So yeah, if you've got an unhealthy river, then you will likely have less mayflies, and that could well affect the swift populations, which is why this project is happening. Has that sort of settled on a number?
It seems to have. I haven't looked at it for a while, but yeah, it does appear to be.
Is it around 8 something?
Yeah, 8. Oh yeah, 8.4.
8.4.
Seems about right. Just went down to 8.3, but I don't know if that's because I moved.
It always does that.
No, it's gone back up again.
It's gone back up.
Yeah, and then the number underneath is that 15.6? I don't know if you need that one.
So that's the temperature.
Temperature, yeah.
That's pretty close to what we got with the
okay,
solid. So that's always good.
That's quite warm, isn't it?
It is quite
15,
warm.
yeah.
Well, I was swimming in it the other day, so during the hot. During
Yeah,
the
yeah,
hot,
yeah.
Not during this kind of. So yeah, it has been fairly warm.
Yeah, this year in general, but a lot of this river, lots of parts of it are quite shallow.
Oh,
And
okay,
that
yeah.
will
So
play a part in
Yeah.
that.
they'll just it's a bit like passive heating, isn't it? It heats up the bottom of the riverbed, and then it'll reflect back in. Yeah.
Exactly.
Okay, is this one done then? Or?
Yeah, I think so. Is it still around the same?
Oh
is
yeah,
it?
it's the same exactly.
Yeah, there we go. So you just tap the sort of on-off button there then to the top one. Give it a little shake and then put the cap back on. Lovely, and then that just goes in there. And then that's the majority of the actual water testing done. So the only other thing then, so we've got our turbidity tube. So this is sort of very straightforward, quite rudimentary really. You just fill it with the water and you'll get an idea of how much there is floating around in there.
Okay, and so you're going to fill that straight from the river this time,
We'll
yeah?
fill it straight from the river, so we get an idea.
A more accurate, because it'll have settled in at the bottom of that bucket to some degree, won't it?
Yeah, exactly. Fill it from that. And we don't expect to get a bad reading from this, but what you do is you look down the bottom, and if you can see the black X,
Yeah.
then it's the turbidity is lower than the highest number on this tube,
I
which
see.
is 30.
Yeah, you keep it still, and I'm trying to move the camera. There we go. Can you see it? Yeah, I can see it.
Yeah, so that's okay. Yeah, you can get longer tubes, which are often quite handy for rivers, because unless you've got either some sort of algal bloom or you've just had like a really heavy storm event, it's probably going to be fairly clear.
Yeah.
But that's all good. That's the main things we test for here. So we just put it as less than 30. There we go. And then what we do, we've got an app to record all this onto. So I'll show you now how it works.
May I hold it for you?
Yeah, thank you.
And then I can show it to these people while you're taking the data.
So,
So you can see the top one
So we've
is
got the
temperature.
Actual app. Okay, cool.
The next one is the
Um,
pH.
yeah,
next
it's,
one
you
down
can
is
get
the
the link from our website.
nitrite,
Um,
phosphates.
So what you do then? Okay.
Oh, there it is, aye. Very nice.
So a crow, I think it's probably being chased off. A little bit too close to a crow's nest. Possibly.
Okay.
go.
So they're the redarts.
Just pop in the name, the date.
So that bottom one is less
And then
than. If you
site.
don't know what that little arrow going, that means.
Temperature is 15.9.
Something like that's less than, isn't it?
That is less than, yeah. 6.0 pH. Nitrate was 0.00.
So you've got name, so location, no grid reference, just.
So because we've got 10 set sites that we're testing at, and we're going to be testing at the same place
Okay.
each time.
Yeah, you know where it is.
Yeah, so we
Yeah,
just
yeah.
have the name, and then we've got the
Fair
grid
play,
reference
yeah.
separately. Total dissolved solids was 33.3.
Thank you for following, Evil Maiden.
Fluid oxygen 8.4. Stability less than 30. Has it rained? Yes, in the last 24 hours. The river level, fairly normal.
Yeah, I'd say normal.
It's not especially high.
Yeah.
And then the only other thing we do then, is we just take a photo or a video up and downstream. So I need to check what the first one was. The first one is upstream. So this is more so if there's anything strange going on with the numbers. we
Sorry.
can compare the photos between readings. And then also if there's a pollution event, you can record that as well using a photo. So we just upload both of those to our app and then we submit it. That's done.
Bread and butter, peanut butter would like to know what type of fish would you find in the river, if any?
So the main ones here, so you've got New Dyfi Fisheries Association help support a lot of the fish life on this river, and they have a lot of trout and salmon. So those would be the main ones. I know there's eels have been seen on the Dyfi as well.
Yep.
Load of lampreys.
Lampreys, yeah.
Yeah, so it's a fairly diverse river in terms of what it supports.
Yep.
Those are the main ones that I know of, at least.
Yeah, sure.
The only other test we're doing, which I don't expect is going to yield very much today, but as part of this project there's a bit of a somewhat experimental element to it, which we are surveying the flying insects as well,
Okay.
in the most non-destructive method we can.
Yeah.
So on our app what we do is we do a 10-minute timed count where we look across the riverbeds and any insects that fly past we count basically. So the idea is that between sites we get
So you
a
don't know
rough
what
idea of-
species it is. You just see something go by, you say one.
Yes.
Yeah, okay.
So
Yeah.
we have on the app, there's the main species that you might be able to identify. So if you spot a bee or a moth, mayflies are a big one. Often you can identify them based on the way they're flying, and if you've got a narrower river. But yeah, for most species it's...
So what if over there and it was a bit more calm and there was a big cloud of midges over there or something? Do you just say midges at like about, I don't know, a metre by a metre? Well, how do you
what we do
quantify
on that,
like
so
tiny insects all swarming sort of thing?
I can show you both now, so if you can see this, just put that in. So this is our form. So we do the time to count, we've got our sites, the type of habitat, we've got our little bit there which sort of just shows you the species that we're mainly counting. And then if you can't see those, you just put other insects there if you're unsure of what it is. And then we have a density. And again, this is a somewhat experimental
plentiful,
method,
yeah,
but
okay.
we have sort of a rough density based on what you can see.
Oh yeah, I see. So
there's a
if you
sort of
can't
key
see
there.
through them, then it's like run away.
Yes.
And
Yeah, you'll know they're there because they'll probably be biting you quite a lot.
yeah. Amazing. So we're just going to stand and look then.
Stand and look for 10 minutes. Do you have
We can do that. Chat. We can look.
When you've got a nicer day than this is quite a nice, relaxing part of the
It could be much
survey.
worse.
Oh, it definitely could. We have had worse here. There we go. Yeah, let's go. Just shout out if you see anything. But anything that's probably won't. But if you see any images, then that's really good. Yeah. I'll put it as a question.
1, 2.
Another one? There's there's one site we test at. There's another one. One site we test at where we are in sort of like a wooded area alongside the riverbank.
One
and
straight over our head.
Another one, yeah. And we've had one day there where we've recorded over 230 mayflies.
Nice.
Which was
Something
quite
small went by
interesting
there.
count. What was it?
Like very small,
Less than 3 mil?
probably. Yeah.
Yeah, so those ones we do as the midge
Yeah.
test account. So this is just the other part of it. It's just you pop in the weather conditions, whether you're in shade.
Yeah.
Everything's been moving all the time. We're quite strongly moving, really. Yeah,
It's a kind of Beaufort scale thing, is it?
Yeah, just so we've got an idea. So as with these sort of the smaller insects like the midges, if we know that it was windy on that day, we know it's not necessarily because of the river. It's the weather.
Yeah, sure.
And the temperature today.
It's 17 now.
17.
It's been 17 all day.
It doesn't feel like it.
No, it doesn't.
There we go.
Okay.
I'm going to head up now to Bardintheyard, which is just up the road, to do the next site.
So,
It's
Bryn,
great to meet you.
thank you very much for today.
Thank you.
It's been really lovely to meet you and James earlier on. James was very knowledgeable, more generally, and it's been really awesome to work with a water treat. Well, you're not a treatment specialist. What is it's water protection and management sort of specialist. Yeah. So yeah.
I'll take that.
Yeah, thank you very much
you very
for your time, and everyone's really enjoyed that. And I think it's something a bit different, and yeah, I might start doing some things like this myself with them now
Good, yeah.
in Scotland as a result of this. So
Brilliant, yeah.
because I have got some of the gear as well, so yeah,
Yeah, definitely. It's worth doing.
yeah, they're all saying thank you very much. Thanks for showing us, doing good works. Thank you very much. Yes, we like this.
Oh, yeah.
So
Well, thank you to everyone as well for coming along. Enjoying.
yeah, thank you.